Hello again, Biology 1103 students, and welcome to our week on nervous systems, the physiology. So the lab this week are going to be looking at neural tissue. And so there's a number of different types of slides. You're going to be looking at pictures of the histology and the tissue itself, and then the cranial nerves and skin receptors. We're going to be looking at the following objectives. We're going to start by looking at a resting membrane potential of a neuron. We're going to talk about how it's generated, how it is propagated, and then how it is transmitted from one neuron to another cell. And we're going to talk about 3 different neurotransmitters. That's your objective, is to name 3 of them. I'll give you more than 3, obviously. So we're going to start by talking about an action potential. Before we start, we need to remember something about the cell membrane. The cell membrane is selectively permeable. So what I'm going to do is I'm going to draw a cell membrane and we're going to talk about this. So you remember that selective permeability, and this is going to be very, very basic. This is our phospholipid bilayer. So each of these lines represents one of those phospholipid one of those layers of phospholipids that gives us our phospholipid bilayer. So remember, when we mean selectively permeable, it means that substances don't necessarily just move freely across the cell membrane. The only substances that move freely across the cell membrane are those very small non-polar molecules, such as oxygen gas, carbon dioxide gas, and sometimes water. This means that the cell can choose what to allow in and what not to allow in. This is specifically, in this case, very important with regards to ions. Ions are small molecules, but they have a charge to them, which means that hydrophobic layer in the middle of our phospholipid bilayer is going to stop the movement of ions through the cell membrane. So the only way that ions can move across the cell membrane is either by facilitated diffusion or by active transport. Okay, so facilitated diffusion, if you remember, of course, only works if there is a high concentration. We're going to say we're talking about sodium ions. There'd be a high concentration of sodium ions on one side of the cell membrane and a low concentration of sodium ions on the other side of the cell membrane. Okay, but even just with that, we need to also have a channel protein on that cell membrane that will allow that sodium to move across. We've got ourselves a channel protein here. I'm going to try to, there we go. I'm just going to, oh, sorry, lost it there. Come on, there we go. Pen, ink color. Let's change that color to blue. Okay, so now I should have a different color. And so what will happen now? I don't have a different color. Didn't work. Okay, so what's going to happen? I'm not going to bother with that now. What's going to happen, since we have a high concentration of sodium ions outside the cell membrane and a low concentration on the inside, then diffusion is going to tell us that in general, sodium ions are going to move from the outside to the inside of the cell. Okay, and in general, they will continue to move until there's an equal concentration of sodium ions on either side of the cell. Okay, but sodium will only move as long as there is a concentration gradient and as long as there is an ion channel open. Okay, obviously the other option is that we can use energy. Okay, and so this is we're going to call this a sodium-potassium pump because this is one that we're going to be using. Sodium-potassium pump. The sodium-potassium pump will move, sorry, let's go back. The sodium-potassium pump will move sodium against its concentration gradient as long as there is energy. Okay, so this is ATP is going to power our sodium-potassium pump. Okay, so we have to remember that ions will only move across the cell membrane if, one, they are pumped across the cell membrane by some sort of pump using active transport, or if there is a concentration gradient and there is an ion channel available to allow them to to facilitate their diffusion. Okay, so we're going to start out by talking about what's called resting membrane potential. Resting membrane potential is shown here. So what we can see is that we have our phospholipid bilayer. We have electrodes that are measuring the electrical potential just outside the cell membrane in the extracellular fluid and just inside the cell membrane in the cytosol. Okay, we have to remember that we are only talking about the area directly next to the cell membrane, okay? All the rest of the extracellular fluid, all the rest of the cytosol can be something completely different, but we are focusing on the area directly outside the cell membrane and directly inside. And we can see that in this case, we have a slight positive charge outside of the cell membrane is slightly positive relative to the inside, or we can put it the other way, that the inside is negative relative to the outside. Okay, so this is a relative term. And in fact, resting membrane potential is when the inside of the cell membrane is negative 70 millivolts more negative, or 70 millivolts more negative than the outside of the cell membrane. So which cells maintain resting potential? Every single cell in the body maintains resting potential. So every cell in the body does what I'm just about to tell you here, okay? But neurons and muscle cells are unique because they do something quite different, okay? And we'll talk about that in a minute. So all cells maintain resting membrane potential, but neurons and muscle cells are unique, and I'll tell you about that in a minute. Okay, so how do we get this slight negative charge inside and slight positive charge outside? So 70 millivolts more negative on the inside than it is on the outside. Well, we do this by moving ions, okay? And by having different concentrations of ions inside and outside of the cell membrane. It's a little bit tricky because what you're going to see here is that we're only moving positive ions, which is kind of weird. But we're going to end up moving more positive ions outside, out of the cell, and move fewer positive ions into the cell. Okay, so in this case, we're just going to accumulate more positivity outside the cell membrane, which means that the inside is going to have less positivity. Okay, it's going to be more negative. So, couple of characteristics that are important. Number one, our sodium-potassium pump, shown over on the right of this image in purple. This is the sodium-potassium pump. Very important. Sodium-potassium pump uses ATP, so it uses energy. This is active transport. It uses ATP and it pumps 3 positive sodium ions out of the cell. At the same time, it's going to pump 2 positive potassium ions into the cell. Okay, so we can see here, we're accumulating more positive ions outside of the cell and fewer positive ions inside the cell. Okay? The second thing that's happening is we're accumulating sodium ions outside of the cell and we're accumulating potassium ions inside the cell. So the sodium ions in this image are shown as the little green circles outside, mostly outside at least. The potassium ions are shown as the little orange triangles that are mostly inside. Okay, the second thing that's going to happen is that we have these leak channels. And the leak channels are a little bit weird because what they do, leak channels are ion channels. So they allow for the diffusion of ions down their concentration gradients. So you can see here that our sodium leak channel is allowing sodium to leak back into the cell, even though our sodium-potassium pump is pumping it out. Okay, this leak channel is allowing it to leak back in. Same thing with the potassium. Our potassium leak channels are allowing the potassium to leak back out. Okay, there's a reason for this. Our sodium-potassium pump requires a constant supply of sodium inside and potassium outside in order to move them in the opposite directions, okay? So in order to move 3 sodiums out, it has to have sodium inside the cell. In order to move the potassiums in, there has to be potassium inside the cell. And they rely on each other. So for example, if we have a whole bunch of potassium outside the cell and we're trying to pump it in, but there's no sodium inside the cell, the sodium-potassium pump will not work. So leak channels allow for the continued movement of the continued active transport of sodium and potassium by that sodium-potassium pump. Okay, The thing is, the trick has to be that the sodium-potassium pump has to pump sodium and potassium faster than the leak channels will allow them to leak. And that's what does happen. What we end up with then is we end up with a high concentration of sodium outside the cell. Okay, we end up with a high concentration of potassium inside the cell. Okay, so those are two critical portions, critical characteristics of our resting membrane potential is a high concentration of sodium outside the cell, high concentration of potassium inside the cell. But we also want to have this electrical potential across the cell membrane. We want to be there to be more of a positive charge outside the cell and more of a negative charge inside the cell. So we've accomplished that by pumping 3 sodiums out for every 2 potassiums in. So we're moving more positive charge out of the cell and less positive charge into the cell. In addition, we have some large anions, negatively charged proteins here, that remain inside the cell and contribute to the negativity inside the cell. Okay, so resting membrane potential is characterized by 3 things. It's characterized by a high concentration of sodium outside the cell, a high concentration of potassium inside the cell, and a membrane potential of negative 70 millivolts. Okay? So this is just going to summarize what I just said, these next slides. So the sodium-potassium pump contributes to membrane potential by transporting 3 sodiums out for every 2 potassiums in. It is pumping more positive ions out of the cell and fewer positive ions inside the cell. So it contributes to the inside of the cell membrane being more negative relative to the outside. We also have the leaky ion channels, okay? So we have sodium and potassium pumps. This focuses on the potassium channels, the leaky potassium channels, because there are more leaky potassium channels than leaky sodium channels, okay? So they're also going to move more ions because there are more leaky potassium channels than leaky sodium channels. There's again going to be a greater movement of positive ions out of the cell than into the cell. Okay? This is going to contribute to the inside of the cell being more negative than the outside. And so the resting membrane potential is due to these 3 factors. The action of the sodium-potassium pump. The sodium-potassium pump does 3 things. It creates the high concentration of sodium outside the cell. It creates the high concentration of potassium inside the cell, and it contributes to that electronegativity, to the fact that the outside of the cell membrane is slightly more positive than the inside of the cell membrane. The leaky potassium channels are the second factor. The leaky potassium channels allow more potassium to leave the cell, and the leaky sodium channels allow some sodium to come in, but not as much. Again, this is going to do 2 things. It's going to power the sodium-potassium pumps to allow the sodium-potassium pump to keep working. And secondly, it's going to contribute to the fact that there is more positive charge outside the cell and more negative charge inside the cell. The third factor is the protein anions. They're negatively charged. They remain trapped in the cell, and they're going to contribute to that negativity inside the cell. Okay? So that's resting membrane potential. There are 3 factors that contribute to resting membrane potential, and there are 3 characteristics that typify resting membrane potential. Okay, so all cells are maintained at resting potential, but neurons and muscle cells are unique because they are what's called excitable. They have special ion channels that open when there is a correct stimulus. Okay, so we've talked about the passive or leakage channels already. I'm going to talk to you about the ligand-gated channels, mechanically gated channels, and voltage-gated channels. Just remember, these are still ion channels. When they open, they facilitate diffusion. That's it. It's all passive transport still. Okay? So we have the passive or leak channel, which opens or closes whenever, randomly. When it's closed, no ions can move across. When it's open, ions will move down the concentration gradient from high concentration to low concentration. Ligand-gated channels Again, when they're closed, they don't allow for any movement of ions. However, when the appropriate chemical or ligand is present, in this case, it's showing a neurotransmitter. This is acetylcholine. We'll talk more about acetylcholine in a bit. When acetylcholine is present here, so this is the correct ligand, it's going to bind to a receptor site on our ligand-gated channel. When our neurotransmitter or chemical is bound to the receptor, it opens the channel and ions will move down their concentration gradient. Okay, we're seeing here that this channel actually allows for 3 different ions to move. It allows sodium to move, potassium, and calcium. They will move in the direction from high concentration to low concentration. So what we can see here is that calcium and sodium both are higher concentration in the extracellular fluid. Potassium is higher concentration in the intracellular fluid. So that's going to determine which direction they move in, and they diffuse in, sorry. Mechanically gated channels open when the cell membrane is distorted. So they're going to respond to things like vibration or stretching of the cell membrane. Anytime the cell membrane is distorted in any way, could be pressure as well, this mechanically gated channel is going to open. Again, when it's closed, it doesn't allow for the movement of any ions. When it's open, it allows for the free movement, the free diffusion of the ions through that channel. Again, we have 2 different ions that are moving here. Sodium is in greater concentration outside than inside, calcium greater inside than outside. When the mechanically gated channel is open, then it's going to allow for the movement of ions. Okay, so the ligand-gated channels, we're going to see this at the axon terminals, and it's going to be involved in the transmission of a signal from a neuron to another cell. The mechanically gated channel, the ligand-gated channel and the mechanically gated channel, we're going to see this when we talk about different sensory receptors. So different sensory receptors are based on this principle here. So some sensory receptors will be based on a ligand-gated channel. Other ones will be operated with a mechanically gated channel. Okay? But the one that we're going to spend a little bit more time on today is the voltage-gated channel. And voltage-gated channels respond to a change in that electrical potential or the electrical charge across the cell membrane changes. So we can see here, this is a cell at resting potential, -70 millivolts. If we see a change, a slight change, this is just getting a little bit less negative on the inside and a little bit less negative to -70 millivolts. What you'll see is the voltage-gated channel will open. Again, allowing for the free movement of ions down their concentration gradient. So, gated channels are specific to specific ions. We saw some examples there where there were multiple ions, and that sometimes does happen, but in general, we'll see gated channels that are specific to one specific ion. And when a gated channel is open, then ions are going to move quickly across the membrane down their electrochemical gradient. So from high concentration to low concentration. Of course, the consequence of ions moving quickly across the cell membrane is that they bring their charge with them. And so if we have positive ions moving quickly from the outside to the inside of the cell membrane, they're going to bring their positive charge with them, and they're going to change the electrical potential across that cell membrane. Okay, this is going to cause something else to happen, as we're going to see when we talk about the action potential. So the action potential is the signal that we talk about when we talk about how an electrical impulse travels along the membrane of a neuron or muscle cell. But just keep that in your mind for when we talk about the muscular system. We're going to focus on the neuron today. And the action potential itself is a reversal of the charge. So we know that at resting potential, we see an electrical potential across the cell membrane of negative 70 millivolts. The inside of the cell membrane is 70 millivolts more negative than the outside. The action potential is a complete reversal of charge, where the inside of the cell membrane becomes more positively charged than the outside. Okay, and the way that this is accomplished, of course, is by allowing a large volume of positive ions to flood inside the cell membrane. And this, of course, they bring their charge with them, resulting in a more positive inside of the membrane compared to the outside.
I've said before that a nerve impulse is an electrical signal that travels along the neuron, but it's not quite electricity as you may know it. Electricity is the flow of electrons, but in a nerve impulse we use the word potential to describe the movement of charged particles across the membrane of a cell. The charged particles flow like a wave down the axon, moving in and out of the cell. The movement is mostly of positively charged ions, not negatively charged electrons like you might imagine. Let's look even closer at the cell membrane. At rest, when a neuron isn't firing, there's a high concentration of sodium ions outside of the cell membrane, and also a high concentration of potassium ions in the cytosol of the cell. The sodium and potassium channels are gated and closed. Because of the difference in number of sodium and potassium ions, as well as the presence of negative ions like chlorine, there's an overall negative internal charge of negative 70 millivolts. The beginning of an impulse is marked by depolarization. This means that the voltage-gated sodium channel is opened and the sodium rushes into the cell. The influx of positive charges quickly shifts the electric potential of the membrane so that now the outside is more negative and the inside of the cell is more positive. This change in polarity will close the sodium gate. Nearby is the voltage-gated potassium channel, which will now open and allow potassium ions to flow out of the cell. The movement of these positive ions will repolarize the cell, allowing the cell to have a negative internal charge relative to the positive extracellular charge. The change in polarity will also close the potassium gate. At this time, you might think that the cell is ready to receive another signal, but it can't. If the sodium gates opened, the sodium ions would not move. In order for ions to move through their channels, they have to be able to move down the electrochemical gradient. The positive sodium ions aren't attracted to the positive environment outside of the cell, so they won't move. Another signal can't be sent until the sodium and potassium ions have moved back to their resting potential positions. But this means moving against concentration gradients, which means energy must be used. During the refractory period, the sodium-potassium pump will move 3 sodium ions out of the cell and 2 potassium ions into the cell with 1 ATP molecule. This must happen constantly and rapidly, so the brain uses a significant amount of your body's energy. In a matter of milliseconds, the sodium-potassium pumps will restore resting potential and another action potential can move through. Nerve impulses are actually waves of action potentials, which means ions are flowing in and out of the cell in a wave-like pattern. Because of the refractory period where sodium ions will not flow out of the cell by diffusion, action potentials can only move in one direction. They begin at the dendrites and they end at the axon terminals. This wave-like region where the action potentials are flowing are referred to as the local current. The flow of sodium through one gate will trigger the flow of sodium in the next gate, but sometimes there isn't enough sodium to trigger an action potential. Using an oscilloscope to display the membrane potential over time, we can see that sometimes the flow of sodium ions isn't enough for an action potential to occur. The internal region of the cell must reach negative 55 millivolts, the threshold potential, for action potentials to begin. If enough sodium ions are released to surpass the threshold potential, an action potential occurs. In the oscilloscope trace of an action potential, we can see where the sodium and potassium gates have opened. Here is the point where the stimulus occurs, and sodium gates open, allowing sodium to rush into the cell. This depolarizes the cell, causing the inside to become positive relative to the outside of the cell. This polarity change opens the potassium gate, which allows potassium ions to rush out of the cell, which rapidly repolarizes the inside to a negative charge. This can often become more negative than negative 70 millivolts and is called hyperpolarization or undershoot. This will be corrected during the refractory period, where the sodium-potassium pump uses ATP to restore resting potential. During this time, no additional action potentials can be sent, but once resting potential is reached, the neuron is ready to fire again. Thanks for watching this episode of Teacher's Pet. Don't forget to like and subscribe and follow me on Twitter @SciencePet.
Okay, so now we watched the video about the action potential, and now I'm going to break it down and talk about the different stages of the action potential. So remember, whenever we're talking about this action potential here, we're focusing in on a single spot on the cell membrane of a neuron, or it could be a single spot on the cell membrane of a muscle cell. So it's the same thing that's going to happen there. But we're focusing on a single location. Okay, so we're going to see all of the stages of what happens at that direct spot on the cell membrane. And then we're going to move on to how that signal passes from one part of the cell membrane to a neighboring part of the cell membrane, and so on down the length of the axon. So we start out, of course, at resting potential. Remember, resting potential has those three characteristics. It's got the high sodium concentration outside the cell membrane. It's got high potassium, sorry, that didn't draw there, high potassium inside the cell membrane, and it's got that membrane potential of minus 70 millivolts, meaning that the inside of the cell membrane is 70 millivolts more negative than the outside. Okay, at this point, our sodium channel is closed and our potassium channel is closed. These are both voltage gated channels, which means that they will open and close in response to changes in the electric potential. When we're at resting potential of minus 70 millivolts, all channels are closed. The next thing that happens is that we get a slight change in the polarity of the membrane, minus 55 millivolts. That means that the inside is still more negative than the outside, but the inside is less negative than it was when we were at resting potential. We'll talk about how we get that slight change in just a minute. So we'll just believe me that there is a slight change in the polarity of the membrane. This causes the sodium channel to open. Okay, the sodium channel actually has two types of gates. It's got an activation gate and a deactivation gate. So the activation gate opens. That's these little side flappy things here. Those are the activation gate. They open when there's a slight change in the polarity of the membrane to minus 55 millivolts. Of course, as soon as that opens, sodium is going to flood into the cell membrane, bringing that positive charge with it. All those positive charges moving into the cell cause a depolarization of the cell membrane, which means, of course, it's a reversal of the polarity from more negative on the inside and more positive on the outside to the change goes to more positive on the inside and more negative on the outside. So we end up with depolarization at plus 30 millivolts. That means the inside of the cell membrane is now 30 millivolts more positive than the outside. Okay, this is shown here on this graph where we have the large peak in our electrical potential here, the large change. Okay, so voltage-gated sodium channels, as I said, it says on the side of the slide here, it says it's an all or none response. Okay, as soon as that threshold polarity is reached of minus 55 millivolts, all the sodium channels open at that point in the cell membrane. Okay, so now we are depolarized. Depolarization, or the change to plus 30 millivolts, causes two things to happen. The first thing that it causes to happen is that the sodium channel deactivation gate closes, shown here. Okay, and the potassium channels open. Potassium is then allowed to flood back out of the cell, taking its positive charge with it. We end up then after repolarization, so after this phase here, we end up back at about action potential, at least about resting potential. We actually end up a little bit hyperpolarized. So we end up at minus 75 millivolts inside, and so minus 75 millivolts more negative on the inside of the cell membrane as compared to the outside. Okay, so we then have, at the end of repolarization, we have returned to the correct electrical potential across the cell membrane. But you remember from the video that we're actually in a refractory phase. And the refractory phase is because the ions are completely opposite from what they should be. At the end of repolarization, you have lots of sodium inside the cell. Okay. And we have lots of potassium outside the cell. Sorry, that was supposed to be an arrow, an up arrow here. Let's just make this into an up arrow. Okay, lots of potassium outside the cell. All right, there, that's prettier. So even though we may be at close to resting potential in terms of our polarity, the ions are in the wrong position. And so this point on the cell membrane is not able to respond to another action potential yet. Because even if we opened up this sodium channel, there's too much sodium inside. The sodium will not flood in like it should. So we need to get back to resting potential before this part of the cell membrane is able to respond to another action potential. Okay, so this is what happens during hyperpolarization. Okay, the sodium channels close when we get to minus 75 millivolts. The potassium channels close when we get to minus 75 millivolts, and the sodium channels reset. So we have this ball here is the deactivation gate. It has to open up, and then the activation gates have to close again in order for this sodium channel to be prepared to accept or to respond to another action potential. In addition, we have to get those sodium ions and the potassium ions back to the correct sides of the cell membrane, and so the sodium-potassium pumps have to take over in order to return back to resting state. At this point, now this portion of the cell membrane is now able to respond to another action potential. Okay, so there are 4 phases of the action potential. We start at a resting state. We remember that resting state happens at minus 70 millivolts. Okay, depolarization, when the sodium gates open, happens at minus 55 millivolts. That's a stimulation for depolarization to occur. Repolarization begins when we get to plus 30 millivolts. This, of course, is the depolarization here. This begins the repolarization at plus 30 millivolts. The signal is sent so the sodium gates close and the potassium gates open. We get to minus 75 millivolts, which is considered to be hyperpolarization. This is the sodium gates reset, the potassium gates close, the sodium-potassium pump takes over, and of course that takes us back to the resting state and minus 70 millivolts. Okay, so what we're going to talk about next is talk about the propagation of the action potential. And this is the fact that an action potential at one point on the membrane will cause an action potential to occur at an adjacent region of the membrane. This is called the propagation of the action potential. So this is what happens. Basically, we have depolarization that occurs at a single point on the axon membrane. That depolarization causes the depolarization of the adjacent segment of the axon membrane. And of course, that means that that next segment is going to then be depolarized. That depolarization at that middle segment is going to cause depolarization at the next segment down. Okay? So remember that this portion right here, being in the refractory phase, is really important because you'll notice that this depolarization here actually can potentially affect the previous portion of the cell membrane. But because this previous portion is in the refractory phase, it's not responsive. The only part that is responsive is the next portion of the cell membrane downstream from the depolarized location. And so we have the action potential that travels, and it will always travel in this direction from the cell body all the way down the axon to the axon terminals. So the action potential only goes in one direction because of that refractory period. Okay? Voltage-gated sodium channels, when they close their inactivation gates, they have to be reset before they can respond to another action potential. Okay, they're still desensitized and cannot open again, which means that the action potentials are unidirectional. Okay, so there are a couple of different ways that the action potential can be conducted. The way that is most efficient is called saltatory conduction, and saltatory refers to jumping, and this is called saltatory because the action potential appears to leap from the space between two myelin sheaths to the next space between the two myelin sheaths. So saltatory conduction only occurs in myelinated axons. Okay, so unmyelinated ones can't do this, but myelinated ones can. So the myelin sheath serves to insulate the axon and allows ions, keeps ions very close to the axon membrane underneath that myelin sheath. It means that there are only voltage-gated channels at the spaces between myelin sheaths, or they're called the nodes. So what happens is that there's an action potential at the first node, and then ionic currents travel in the extracellular fluid surrounding the myelin sheath, and then the action potential is then generated at the second node. So all you need is enough of an ionic current to travel underneath the myelin sheath from one node to the other in order to cause a slight change in the electrical potential in that second node to minus 55 millivolts from minus 70. Remember, it's not very much of a change that needs to happen. So, of course, the advantages of saltatory conduction are that it happens faster. Okay, so it's much faster transmission of the electrical signal, and less ATP is used. So that's something important to remember, is that whenever we talk about the action of the sodium-potassium pumps, pumping sodium and potassium ions across the cell membrane, every time that pump works, it requires a molecule of ATP. The fewer action, at least the fewer spots on the axon membrane that actually become depolarized, then that means that there's less of a requirement for ATP in order to repolarize those that portion of the cell membrane. Okay, so I'm just going to demonstrate to you how ionic flow works. And in order to do this, I'm going to draw the cell membrane here. Okay, and just so you can get the sense of what's going on, I'm going to draw the other side of the cell membrane. Okay, so this is the phospholipid bilayer here. I'll draw both layers of it just to keep you oriented, and then this is a phospholipid bilayer on the other side of the axon. Okay, so we have the intracellular fluid here that's inside our axon, and we have the extracellular fluid out here, and of course extracellular fluid, let me see if I can spell properly, extracellular fluid out here as well. Okay, so here is the inside of the axon. So what happens, of course, is I'm not going to draw it on both sides of our axon. I'll only concentrate on one. But of course, when we are at resting potential, we have a slight positive charge on the outside of the cell membrane. So we'll just go along here and draw this. Okay. Positive, 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 positive. We've got a slight negative charge on the inside of the cell membrane. In fact, it's minus 70, of course, is our resting potential. Okay, so what happens is that, let me see if I can get this to change to a different pen color so that we can see what's going on here. All right, now it's blue. That's awesome. Okay, so what happens, of course, is when we have an action potential is that we have a reversal of charge. So when we have a reversal of charge, of course, what that means is that all of this becomes negative on the outside relative to positive on the inside. So we've now had an action potential at this point in the cell membrane right here. Okay, that point there is now depolarized. Now, the point is that we know from experiences of electricity that positive and negative charges are attracted to one another. So these positive charges that flow into the cell, okay, so all these positive sodium ions that have flowed into the cell are not going to stay here. They're going to travel. Again, I'm going to try to change to a different color here. Just going to have to bear with me a little bit. Let's see, let's use yellow. Did I do that? Okay, so these positive charges are going to flow towards the negative charges on either side of them. Same thing with these negative charges out here are the negative charges that accumulate near the cell membrane when the positive charges leave are also going to flow in one direction or the other. Okay, so this is ionic flow. And what that means is that, of course, when we have ionic flow, is that our negative ions flowing to the right and our positive ions flowing to the right will result in a slight change in the cell membrane action potential at the next location, okay? Because there's going to be a little bit of positive charge mixed in here and a little bit of negative charge mixed in here. And this will cause another action potential to occur at this location on the cell membrane, okay? And of course, what will happen again is that our ions will not stay in the same location. They'll flow away from where they moved, and they'll flow in both directions, of course. I'm just going to make a point of this, though. They're going to flow in both directions, but, of course, the important thing to remember is that, change to this color here, these, of course, this part of the cell membrane that has just been depolarized is not responsive. So even though the ions are flowing to the left here, they're not going to cause an action potential again in the same portion of the cell membrane that was just depolarized. Okay, it can't do that. But we can, of course, result in a slight change in polarity because of the mixing of the ions here that will result in a large change of polarity and an action potential occurring here. Okay, and then we continue on. We can keep going and we can say, okay, now there's been a slight change in polarity. The ions are going to flow, and they'll flow in both directions, of course. Okay, but the ions will not be able to affect a change in the part of the cell membrane that has just been depolarized, but it will cause a slight change in the polarity because of the ionic flow, slight change in polarity downstream, which results in, of course, a large change in polarity, the action potential at this point. And we can continue on all the way down the whole cell membrane. Okay? Now, what we can do then, sorry, I'm going to try to take away not a lot of the stuff that I wanted here. I took away one of my cell membranes, though, so I didn't really want to do that. Let's see. Take away some of this stuff so I can show you. Okay, so I'm just going to leave that here. I'm going to go back to my red pen. Red pen. I'm having trouble getting the right color here. Okay, my red pen, and I'm going to draw. Oh boy, that's a nice thick cell membrane. Oh well, there we are. Got a thick cell membrane. I did not want this to be the highlighter, though. It's the pen. Okay, so we've got our cell membrane back. So now what we're going to imagine is we're going to imagine that we have a myelin sheath surrounding this cell membrane. I'm going to do the myelin sheath in purple. And so the myelin sheath kind of looks like a hot dog bun on either side of the cell membrane. So this is the myelin sheath. This is the same one on either side. And what happens, of course, is that with a myelin sheath, we don't have, underneath this myelin sheath, there are no voltage-gated channels. There are voltage-gated channels just before. So I'm going to put another myelin sheath here that kind of extends here. So there will be voltage-gated channels in this area here. Okay, so the area between the two cell membrane, between the two myelin sheaths at least, there will be some voltage-gated channels. And so what we're going to see is that, of course, we have a slight change in polarity that comes along. And the slight change of polarity, we're not going to talk about how it gets there, but the slight change in polarity is going to result in an action potential here underneath in that gap between the two myelin sheaths. Okay, again, same sort of thing happens. We have the flooding in of positive ions to the inside of the cell membrane and leaving negative ions on the outside of the cell membrane. So those negative ions at the outside of the cell membrane will want to flow towards the positive ions. Okay, and so they're going to flow underneath this sausage here. Again, the positive ions, similarly, are going to flow along the inside of the cell membrane, but it's really the negative ones. So what we're seeing here is that instead of having these negative ions that are gathered at the cell membrane within the node, rather than having them just flow off in all sorts of directions, right? Rather than that, what the myelin sheath is doing is the myelin sheath is going to make sure that the ions don't flow off in every direction, that the ions travel underneath that myelin sheath all the way to the next node, where there are, once again, of course, in this next node, there are going to be voltage-gated channels, and these ions are going to mix in. The positive ions are going to mix in with the negative ions inside. The negative ions are going to mix with the positive ions on the outside, causing an action potential at this next node. Okay, so that's how ionic flow works. Hopefully that will help you to understand what was going on with the myelin sheath and saltatory conduction. Of course, if there aren't any myelin sheaths, then we have continuous conduction that occurs. Okay, so this is kind of what it looks like. So in the nodes, of course, there's depolarization. The flow of ions occurs underneath the myelin sheaths and then results in depolarization at the next node. So what happens, of course, is that if you have a very short axon, so for example, these dendrites, the dendrites, remember, are the projections coming from the cell body of a neuron. In the dendrites, usually there are no voltage-gated channels. So basically the stimulus occurs and the voltage decays. You can see it's decaying as it goes all the way along. But that's okay because it can persist for long enough to result in an action potential as soon as it does get to a voltage-gated channel. Along an unmyelinated axon, we have this continual renewal of our action potential down the whole length of our axon. And then, of course, that's continuous conduction shown in B here. In C, we see saltatory conduction, and you can see that the insulation of the myelin sheath results in lesser decay of the signal over a longer distance. So we don't need to open and close nearly as many voltage-gated channels, and that means that we don't need to restore resting potential in as many points along our axon. And that, of course, means we're not using as much energy in order to propagate the signal along the same distance. This is really important for some really long neurons that we have in our body, especially some of these spinal nerves are incredibly long. Okay, so finally we get to what happens when we get to the end of a neuron. We get to the end of the axon, we get to the axon terminals. We want to pass that message on to another cell. Can be passing it along to another neuron, as shown in this image here. We have a presynaptic neuron, and then we have a postsynaptic neuron. So the presynaptic neuron is the neuron that has the message right now, and it's passing it over to the postsynaptic neuron. Instead of having a postsynaptic neuron, you could have a postsynaptic muscle cell, or you could have a postsynaptic gland as well. So a couple of stages of the events at what is called the synapse. So the synapse is actually the place It identifies the place where the presynaptic neuron's axon terminal comes close to the membrane of the postsynaptic neuron. Does not touch though. You'll notice that there is a small space called the synaptic cleft. So we have a couple of events that are required in order for the message to be transmitted from one cell to the other. First of all, we have to know what's going on here. In the axon terminal, in the terminal bulb, there are a couple of important characteristics. The first one is that there are voltage-gated calcium channels. Okay, these are voltage-gated calcium channels, not sodium channels anymore, not potassium channels. These are calcium channels. We also have to understand that calcium is far more abundant in the extracellular fluid as compared to inside the neuron. Secondly is that there are synaptic vesicles in the axon terminals, and these synaptic vesicles contain a neurotransmitter. It can be different neurotransmitters depending on which type of neuron it is. Okay, so in addition, across the synaptic cleft on the postsynaptic neuron, there are ligand-gated ion channels, and they can be different types of allowing the movement of different types of ions depending on the function of this synapse, depending whether it's excitatory or inhibitory, or whether it's associated with a neuron, or associated with a muscle cell, or associated with a gland. Okay, so the stages of the events at the synapse. First of all, the action potential is going to travel down our presynaptic neuron membrane and arrive at the axon terminal. When it arrives at the axon terminal, it depolarizes the axon terminal, and it results in the opening of the voltage-gated calcium channels. Okay, so the depolarization of the axon terminal opens voltage-gated calcium channels. Calcium floods into the axon terminal. Calcium entry, as shown over here, it causes those synaptic vessels, it binds to the synaptic vesicles. Those synaptic vesicles will then move towards the presynaptic membrane and release their contents into the synaptic cleft. Okay, so now we have the release of a neurotransmitter into the synaptic cleft by the presynaptic neuron. In the postsynaptic neuron membrane, remember, we have ligand-gated ion channels. Remember, ligand-gated channels open or close depending on whether a specific chemical is present and bound to them. In this case, these are going to open when the neurotransmitter is bound to them. So we have binding of the neurotransmitter to a ligand-gated channels. This allows an ion to move across the postsynaptic neuron membrane. Again, depending on whether it's excitatory or inhibitory, you might have movement of ions in one direction or movement of ions in another direction. It doesn't really matter. It's going to cause ions to move. Okay? So we've now, when we have the movement of ions in the postsynaptic neuron, this could generate another action potential that will be propagated down our postsynaptic neuron. It's also possible, if it's an inhibitory neurotransmitter, it will stop. It will reduce the likelihood of an action potential being propagated along our postsynaptic neuron. But that's a detail that's not that important for you guys to remember, at least to fully explain in this case. Okay, the only thing is that we don't want those the ligand-gated ion channels to remain open forever. So we have to have some mechanism for removing our ligand or our neurotransmitter in order to allow them to close and allow our postsynaptic neuron to go back to resting potential. So this is what happens here, is that we have, of course, the binding the neurotransmitter has opened these ligand-gated ion channels, and you can see here that the ion movement goes in either direction. Now, in order to allow our postsynaptic membrane to go back to resting potential, we have to close these ligand-gated ion channels. And we have to do that by removing the neurotransmitter. And there are three ways that the neurotransmitter is removed. First of all, sometimes just diffuses away, of course, because the neurotransmitter is in high concentration in the synapse, in the synaptic cleft at least, and so it's going to tend to diffuse away from where it's highest concentration to where it's the lowest concentration. There are enzymes that will break down this neurotransmitter. We're going to see this when we talk about muscular excitation, is that the neurotransmitter acetylcholine is removed by acetylcholinesterase, which is an enzyme. We also have reuptake by the presynaptic neuron. So the presynaptic neuron can take the neurotransmitter back and can reuse it again. Regardless, once we have no more neurotransmitter in the synaptic cleft, our ligand-gated channels in the postsynaptic neuron close, and then that, of course, stops the signal until, of course, another signal comes along. So some examples of neurotransmitters. There are 30 different neurotransmitters that have been identified. Each of them do have a specific function. A neurotransmitter can be inhibitory or excitatory, or both, depending on where in the nervous system it is being used. And certain neurotransmitters are used by different neurons responsible for a particular function. So we're going to talk about acetylcholine first of all. Acetylcholine is released at many synapses in the brain and in the peripheral nervous system. So it's used throughout the nervous system. We're going to focus on its use at the neuromuscular junction. This is between a neuron and a muscle cell. It is removed by acetylcholinesterase, which is an enzyme. Depending on where it is used in the nervous system, it can be excitatory or inhibitory. When it's at the neuromuscular junction, it tends to be excitatory, which means that it's going to cause an action potential in the muscle cell membrane. These are some other important neurotransmitters. And just make sure that you can recognize a couple of them. There are ones like glutamate, which is the main excitatory neurotransmitter. There's GABA and glycine, which are the inhibitory neurotransmitters in the brain and the spinal cord. Dopamine, of course, is a neurotransmitter that's involved in motivation. Norepinephrine is involved in the sympathetic response. Serotonin is involved in satisfaction. Acetylcholine, of course, muscle contraction there. And then we have some endorphins, enkephalin and endorphin, that, of course, have analgesic pain-reducing and pleasure-inducing effects. So you should know a couple of these. Definitely know acetylcholine and then maybe 2 or 3 more of them. Okay, so that's the end of our discussion of the nervous physiology. Next week, we'll be focusing on some of the special senses. In the lab, you'll be looking at the eye and the ear, which is kind of cool because you get to do some tests on yourself and see what it is. I will see you all on Thursday, and I hope you have a great week.